thank you so much, and it's a wonderful introduction. Maybe I should quit. and thank you for being here. It's I think I was here a few years ago, and it's always great to be here and see the progress, tremendous progress over the last few years. You guys are a bioengineering department in steroids. I mean, you guys are growing faster than any place I've known. And in a very nice, balanced way and big breakthroughs. So it's a pleasure to be here with colleagues and friends. It was about 1984 or 85. I was at MIT student, and my advisor, I was doing low temperature biology. That's still something I do. And my advisor's advisor, my advisor was from UC Berkeley, Ernie Cravallo, and his advisor, so my academic grandfather, came to MIT, and so I got, at the time he was the chancellor of UC Berkeley, very famous heat transfer thermodynamics professor who's really considered to be one of the first who got into nanoscale processes and events in heat transfer and thermodynamics. He was very famous scientific, his scientific work was very, very well known, Professor Tian. So I had to have a lunch with him. And one of the things he told me is I'm trying to emulate as much as I can. He said, Mehmet, go to extreme, you will discover interesting things. So that really stuck with me over the next 30 years or so. And many of the time we try to go to extreme. We don't always succeed, but I love going to minus 196 degrees Celsius with living cells and bring them back. And we were last night talking about I want to build a colony in the lunar South Pole for all mammalian species germlines, because it's at cryogenic temperatures for the last few billion years. It doesn't see the solar. So it's going to that extreme. And part of that, about 10 years ago, and we were doing microfluidics, and I was told, nah, microfluidics, you can, the fame comes from processing small amounts. You can't do big amounts. It's whoever does smaller amount is what's important. And but I was in a hospital, and I want to get into more applications in blood and diagnostics. So we said, can't we use microfluidics in an extreme way, where I call it putting a garden hose through the chip, a microfluidic chip, where you have to process whole blood, 10, 20. In fact, we have technologies now. We could even do liters of bodily fluids through these channels. And so we started looking, what would be an exciting application? And as I started actually reading the literature, circulating tumor cells came up a lot. And not in my context of microfluidics, but people have been working on circulating tumor cells, and they were using some of the old technologies to find them, not so successfully. And I thought this might be a great opportunity, and again, an extreme science in the sense that these cells are 1 in 10 billion blood cells, so they're extremely rare. And so we started thinking about it and going to Blackboard and designing ideas. And those are the best, most fun part of the research, actually, the early phases of when you really don't know what you don't know and you're excited about it. And then you realize it's pretty difficult. And more recently, we got into clusters, so it's even rarer than rare cell. And so what I want to do today is just give you a sense of the technologies we developed and some biological applications, not a lot of the clinical stuff today, and but some of the biological applications. Like anything, nothing we think is actually original, or nothing I think is original. What Rashid thinks is always original, but this goes back a few millennia. And Hippocrates was, and later Dr. Galen, very famous Roman doctor, and of course you all know Hippocrates. They defined all diseases in 4 fluids, and they called them humors. There is the phlegm, blood, yellow bile. Those are, make sense. And then there's the black bile. Black in Greek is melan, and bile is cholicus, so melancholicus, and it was given for 2 diseases, cancer and mental diseases. And that's why that guy looks, of all of those, it's the most unhappy guy there. And the reason why they called it black bile, at the time, cancer was diagnosed very late. And typically breast cancer, and it's called fungating or wounding cancer because it will break out of the tissue into the skin, and it will look black. So they thought there was a black fluid circulating, causing cancer. And, but of course, Romans didn't do autopsies, so they couldn't really do it. They did some animal experiments. They couldn't figure it out. But then comes Vesalius, very famous father of modern anatomy in 1500s, and he started doing the first extensive autopsies. And he was coming from the school of Dr. Galen and believed in the four humors of disease processes. So he's done a lot of these autopsies, but he was never able to find the black bile. And so he was very disappointed. He published his results, and then life moved on. And a few more hundred centuries later, a resident at the University of Monash in Australia has at the observation post-mortem a 38-year-old male cancer patient. And you can see Dr. Monash sitting there in the laboratory. He was the resident at the Monash University, and he published a wonderful one-page article in 1869. And this sentence is from that. The fact of cells identical with those of the cancer itself being seen in the blood may tend to throw some light upon the mode of origin of multiple tumors existing in the same person. And this is pretty visionary for the time. And today we know that 7 million people die from cancer every year. We know that 1 in 2 men and 1 in 3 women in their lifetime will be diagnosed in this room with cancer. So the numbers are pretty nasty. And we know that 9 out of 10 deaths in cancer is because cancer spreads. Primary tumor very rarely kills. So we know it goes. We know there's a black bile. And he showed here in the pathologist images. This is the primary tumor. You can see the nucleus. You can see nucleoli. This is the peripheral blood sample. You can see 3 cells that look exactly like the tumor cells, and those are the circulating tumor cells. So it's since following almost 150 years, we learned a lot about circulating tumor cells. And what happens is primary tumor forms. As soon as it's the size of a few hundred micron, it's vascularized. And then these cells start going into homeostasis with the body. They are sloughed off from the primary tumor, and they go. And once in a while, it's like a zip car. Once in a while, they can find the right niche, nice location, and they don't die. They home to that location, and they grow, and they colonize. So this process now is well known. It has 20-some steps, and that are very critical in the intravasation and extravasation of these cells. And imagine if you could find these cells in circulation from a blood test, which is minimally invasive. You do it every year, multiple times when you go to your doctor. You can imagine the ways you can personalize the treatment of cancer. You can diagnose it, you can monitor it, you can look at the changes, whether or not somebody is responding. So it would be a very powerful tool, the bioassay, that could change the way we deal with cancer. Almost like what we do in infectious disease, you could turn it into what we call a chronic disease because you can monitor it very closely. And now in cancers, we have so many different drugs. These decisions are far more important than 20 years ago. Time is of essence. And so the presence of these cells are known. The biology of it we know, but we can't find them. And so let's look at some numbers here. If you take a tube of blood, it is 10 milliliters of blood. You have 5 liters of blood in your body. That's 10 milliliters. There are about 50 to 80 billion cells in there. And there are typically, in the technologies I showed you, we can find 1 tumor cell in the tube of blood. So it's a sensitivity of almost 1 in 80 billion. And so if you were to look at each cell for 1 second. Blood cell, tumor cell. Do you know how long it will take you to look at a tube of blood? Any guesses? Days, weeks, years? Years. Years, sold, gone. Any other? 3,470 years. And so if you take once again, the technologies, the last one I will show you is we interrogated at about 50 million cells per second. And so it has to be fast. If I tell you in the entire world there are about 7 billion people, that's 1 milliliter of blood, 1/10 of the tube. If I tell you there is one guy with 3 ears or 3 eyes and go and find it, that's a pretty challenging task. And so it is, I'm just trying to get you to understand that these rare cells that are very important. So people said that they are not in the circulation, immune system removes them, complement removes them, it's not useful. Maybe you find it 10% of the time in metastatic disease, then it's clinically not exciting. So, and people used mostly, I won't go into details of it, technologies that are based on size, density, biophysical parameters, differences, bulk differences, but tumor cells vary. Or they took magnetic sorting techniques or flow cytometry, tried to apply to a rare cell, technologies developed for common cells and try to apply it to rare cells. The way I look at it, or I looked at it 10 years ago, I said, look, these guys are trying to race in Formula One with a Hyundai. You can't win that race. You need to build a Ferrari equivalent of it. And then we built the first CTC chip, circulating tumor cell chip. And that was in 2007. And it made it into this review article by Pantel. And so I'll go through some of the design principles quickly because I want to get to the 3rd generation chip. That's what, and the 1st generation, this was a micropose. It was about the size of a credit card. It was etched using deep reactive ion etching and silicon. And you see the posts. Posts are about 40, 50 micron diameter, 100, 120 micron tall, very nicely manufactured. And we covered the post with an antibody. 85% of all neoplastic diseases, cancer, is in epithelial cells in different organs. So we covered this with an epithelial antibody, EpCAM, epithelial cell adhesion molecule. All epithelial cells have these antigens on their surface. We put an antibody. It's like a very precise immunoaffinity chromatography. There are hundreds, thousands of these posts. And the flow rate here is about 2 milliliter per hour. So it takes about 4 or 5 hours to process a tube of blood. It interrogates about 1 million cells per second. Each post every second interrogates only 10 to 20 cells. So you make them by 100,000 posts. They're I won't go into fluid mechanics. They're distributed in a clever way and to break the streamlines. And the whole blood goes through this, antibody coated. And here is the first lung tumor cell we captured in our first patient experiment. In the next slide, I'll show you the inner working of the device in a video, if it works. Half of the time it doesn't. And whole blood will go from your left to right. You won't see the whole blood because we didn't label it. You have to take my word. We spiked in lots of tumor cells in it. It's not rare anymore because we don't want to wait 3 hours here to see a cell. And so that you see the flow of conditions and how it captures. You see how it goes around and once in a while they stick. There are 5, that one is stuck there. I think this one gets caught right there, or, yep, that one. And they stick all around it because it's laminar flow. And there's some flow of separation in the back, but it's not that in these flow rates, it's not that critical. It's shear stress is very low in these devices. We don't push it very hard. And that's a very straightforward idea. What this did is we did a targeted therapy study published in New England Journal of Medicine. We showed that we can get these cells very routinely in metastatic cancer patients, in lung, prostate, and breast. So it kind of changed the dogma, the belief that they didn't exist in circulation in enough numbers. And that started a whole group of investigators developing technologies. The problem with this technology was expensive to manufacture at the time. So we wanted to see the blood, because surgeons love to see the blood as it goes through. And so we designed the second generation, which is based on herringbone structure. In this case, we don't have posts, but what we have is a flat bottom, and the top surface has these herringbone structures originally designed for molecular mixing by Whitesides. Basically, you get this chaotic advection mixing patterns. But this could be manufactured in plastic. And the Hollywood picture, that the embarrassing picture Rashid showed, came from our funding from Stand Up to Cancer. It's a Hollywood organization that's a philanthropic organization that funded our team as a dream team to do a large-scale clinical trial. We just finished that. I won't go into details of that. But we could manufacture this chip now in plastic, controlled in reasonable amounts. We could put it in the hands of other people to test it. And in the next video, you will see the inner working of that. It will be a little bit different because the cells will be tumbling, so they'll be in and out of. You see how they come and they stick to top and bottom. That's, again, antibody coated. It's positive selection. So as we have gotten into this, we realize the tumor is smarter than we think. And this is a gallery of images from a number of patients. First of all, look at this. This is same lung cancer patient. Green cells here are cytokeratin, so those are tumor cells. Blue is the nucleus. CD45 is common leukocyte antigen, so that's a white blood cell. Tiny. Look at this. That is like mega size. But in the same patient, look at this tumor cell. It's smaller than leukocyte. And morphology is changing. It's same in prostate cancer. Then we looked at the phenotype of these cells. Some of them were dividing, proliferating. Some of them we even got it as it's dividing into two cells in circulation. Look at it, it's in telophase. Some of them are in mitosis. Many of them are going through apoptosing, so they're committing suicide because these are epithelial cells. They're used to sticking to surface. Once you take them off the surface, they don't like it. They commit suicide. It's called anoikis. And there's always a Greek word for every biological process. And apoptosis is committing suicide. Some of them are covered with platelet, so they are stealth. Some of them go through what's called EMT, epithelial to mesenchymal transition. I'll talk about it because that's a very important fundamental issue in cancer. Some of them come in clusters. I'll talk about that too. So what we realized that they come like Ben & Jerry's ice cream in multiple different flavors. And positive selection of going just after EpCAM, and you need to express it high level. Low EpCAM, you miss it. No EpCAM, such as non-epithelial melanoma, no marker. How do you find melanoma cells? There's no marker for them. If they're covered with platelet, you don't get it. So we had to come up with an idea. Here we are looking in the needle in a haystack, and with positive selection based on antibody against tumor surface characteristics, you're trying to go and find the needle from the haystack. So we said, can we turn this around? Can we find all CTCs without even knowing if the person has cancer or what type of cancer? And so independent of their size and phenotype. Of course, if you can go after the haystack, you could do that, right? If you go and throw the haystack in garbage, you're left with tumor cells. But your problem now in a tube of blood, that's what we process, about 200 billion cells. You need to throw away 199 billion, 999 million, 999,999 cells without losing that 1 to 10 cells. As you know, when the fluid moves, of course, that rare cell is going to go to garbage with the rest of the haystack. So how do we get around it? What would you do? At the time, very bright, now he's a faculty member at UCLA, postdoc. I think you overlapped with him, Dino Di Carlo. And we were doing some experiments. And the idea was, we were pushing fluid in microchannels a little bit faster than the average guy. Because it's always laminar flow, Reynolds number of less than 1 in these microchannels. We were going to 5, 10, 20 Reynolds numbers, still laminar flow. So you see one channel here, you see the cells. And we're going to increase the flow rate. You see, if they come in the streamline here, they of course go in that streamline. If in that, they don't switch streamline. That's laminar flow definition. Otherwise, the physics will be totally out of the window. Look at it. We increase the flow rates. Now they started actually being in only 2 lines. They shift, and they find 2 locations that are equilibrium locations. I won't go into details of it, but what happens is when the particle size is the same order of magnitude as the channel, the wall effects, as well as the shear gradient that the particle sees, pushes towards the wall pushes it back, and they find these beautiful equilibrium positions. And so we looked and said, oh my God, particles are behaving independent of each other. If I want to throw billions of cells in garbage without affecting if this is a tumor cell, that's a white blood cell, I want to throw this in garbage, I don't want this to go with that. This is all particles, and it's high throughput. We can put billions of cells through these channels within minutes. And so we said, OK, can we turn this into a technology that could find rare cells? So what we said, red blood cells, platelets are small. We could make a mechanical filter for those. White blood cells are in the same size as tumor cells. So we added microbeads to blood, tiny magnetic beads, coated with antibodies against white blood cells. Those reagents are very well known. They don't change. So it's not like the tumor thing. They don't change. We know the reagents. We know the antigens. And so we mix the blood with microbeads and a bunch of different antibodies. And the first part, it has a 3-stage process. The first part is a mechanical filter. It's a pachinko device, like a Japanese game. You drop the money, small money goes straight, bigger money bangs into walls and goes to the left. And originally described by Dr. Austin over at Bob Austin at Princeton. We scaled it up to whole blood and so on and so forth. It's called deterministic lateral displacement. So whole blood comes, buffer comes here. Red blood cells, platelets, and free magnetic beads come out to garbage. All the nucleated cells, white blood cells and tumor cells, are deflected in the pachinko. Then they come to the Dinos inertial focusing device. By playing with the geometry of that, I won't go into details, we can focus them wherever we want to. And so we line them up. And then they come to the magnetopheresis system. Because now they are independent of each other, you can magnetically deflect these cells. Because it's so well aligned, all you need is to deflect them 5 micron, you separate it. So it's an extremely high throughput, extremely sensitive, and every particle behave independent of each other. It's easy because you pay $500, your artist makes this beautiful figure for you. And it really looks nice, and it makes cover pages and good on your walls. But you need to make it. So this is the final chip. It went through 3 years of evolution. Parts of it was each part made in different materials and different ways, and then we combined it. I won't go into the history of that. And this is done on a CD because Sony makes it. We didn't want to change their form factor. It doesn't spin. And it's symmetrical. This chip processes about 20 mil blood in an hour, and whole blood. And it sorts about 50 million cells per second. Whole blood comes here. That's the pachinko. There are 2 million microfeatures in there. This is the world's most complicated plastic manufacture. The cost now is probably $20, $30. About 2 million microfeatures. There are 256 pachinko devices there, micro devices parallel, and another 256 here. Red blood cells go to garbage. All these resistances to adjust the flows and so on and so forth. This is the first inertial focusing area. This is the most critical part of the device. Then it comes into the magnetic region. There are magnets under this. And I'll show you later why. We inertially focus it again and then magnetically deflect twice. And we collect the rare CTCs here. Red blood cells junk goes out of here. White blood cell junk goes out of there.
What's that third serpentine region, the small one?
Which one? This one?
Yeah.
That's just the resistances to adjust the flow. So that as we put the complexity onto the chip, the peripheral complexity became very simple. So the processor didn't have all the pressure controls that required. So it evolved very fast into a chip that has almost, we have actually, the new versions have even more complexity, but then essentially this is the way it works. I won't go into too much of the details of this, but just to give you, we put green and red labeled, white blood cells, red blood cells, and then to see if we can, show you. This is one of the 256 pachinko devices. They come together, red and green. As you go downstream, that's waste. Red cells are separated from white blood cells. White blood cells go into inertial focusing, randomly distributed in the channel. Then it's a single streak image. These are streak images of the fluorescent images into a single file. And this is the magnetic deflection, single file. We make it 2 to get the high throughput faster. And then you can see in the magnetic region, white blood cells collected, CTCs go to collection. And this is a video of the, again, this is, we spike many cells so that we can see during the time period I'm giving the talk. And if you are crazy, come over to the lab, wait for 2 days under the microscope, you can see one of these. And these are white blood cells coated with beads. This is the earlier version. The problem we had, the processing magnetic field was so sensitive that even if we had one bead on a cell, we would move it to the wall and it would clog. That's why we did the version I showed you that has 2 magnetic circuits. The cells come in from here, stabilize here, inertially focus here. The first one is a low magnetic field. So we separate cells that have 7 to 8 to 10 beads on their surface. They move about 50 millimeter per second, and we have a separation about 10,000 cells per second. And this is after RBC platelet removal. And then here is the second part of the inertial focusing. This is the high magnetic field. In that, our sensitivity is actually half a bead. We can remove that cell. So it's a very sensitive. So that's where the rays, they go around twice. And then you worry about wall-to-chip coupling. If you're doing things in the hospital, your colleagues are not engineers. They want to use this. If you don't give them something usable, they yell at you. And so with Johnson & Johnson, we are manufacturing this to a point where it can be scaled, and we are running lots of clinical studies. J&J is doing 5-center clinical trials in this year. So this black, ugly plastic is just to put all the lines in and out. And there's a processor. The chip sits right here. Blood is here. Buffer is here. That's waste, red blood cell, waste, white blood cells. And then here in the middle, you collect the circulating tumor cells. They come with some other contaminating cells. There are other progenitor cells, fibrocytes, and some white blood cells. Typically, in 1 ml of blood, we have 200 to 400 cells within which there are tumor cells. I love this picture because this is a melanoma, so you couldn't capture these cells with positive selection. And it's almost 250 years after Ashworth's original study in 1869, where he showed the primary tumor. This is fine needle aspirate. That's the primary tumor. That's a melanocyte right there. You see the nucleus, you see the nucleoli, and that's in the peripheral blood. And so that is, for the first time, this is clinical levels diagnostic quality cytology. You couldn't do this with the other techniques because here cells are in a tube. We could culture them. We just published a paper. We can do standard cytology. We could do single cell sequencing. So because they are isolated, pristine, live cells in a tube, we could do anything we want to do. And we have done everything that you can imagine, of course. But what I want to do in the remaining time is to talk more about the mechanism of metastasis. But first, I want to show you that the technology, as I promised at the beginning, separates these cells independent of size and EpCAM, or surface antigen, tumor antigen. We have done this with thousands of cells now, with real patients. These are not cell lines. We measured the EpCAM expression on the surface of CTCs, and as we defined by a number of markers, and cell diameter. You see that these are white blood cells. Many of them are in the same range. So size separation is you're going to lose most of your cells. If you look at EpCAM expression, this is typically, this is the region where you could capture with positive selection. So all these cells on this side you will be losing. And so it shows that we can capture a very general phenotype and size of these cells. And then we have done mass flow cytometry of these cells. This is just one example from prostate cancer to look at 50 different protein markers on these cells and show that many cells that don't have EpCAM actually has PSA, PSMA. That's a marker for prostate. So again, you wouldn't be able to get these cells. Many of the cells that have EpCAM don't have the other markers. So it is very heterogeneous population of cells. And so that this is the general technology. So what I would like to do next is talk a little bit how we can use tools like this to understand biology of metastasis. One part is clinical use. The other, maybe more exciting part is you can now use these tools to understand the biology of these cells. There are two overall hypotheses for metastatic cascade. One is put forth more recently by Bob Weinberg at MIT called epithelial mesenchymal transition. Epithelial cells go to mesenchymal phenotype. They become more motile, more aggressive. They slough off from the primary tumor, circulate, and cause metastatic process. The other one, which is much earlier by Fiedler in 1950s and 60s, they called it tumor emboli, embolus. And they believed that that was clusters of cells slough off from the primary tumor, not single cells. And they get clogged in capillaries because they're fat big, and they grow into the capillary. And so that was a much more mechanical theory. So we wanted to go after the mechanism of the process. Now we have a tool to do that. The EMT has been shown in animal models, but has never been shown in human cancer patients. So we have done a breast cancer study. We have developed epithelial markers, 7 markers in green, and mesenchymal markers in red, 3 different markers mixed. And this is RNA-ish, so we are staining for RNA. There are no good antibodies for this, and it took a long time to optimize all this process, of course. Green is epithelial, red is mesenchymal. This is the same breast cancer patient. Look at it. Some cells are all green, some cells are all red, some are in between. So this is the first demonstration of epithelial mesenchymal transition in an actual patient. Here is an index patient. That's so amazing. And look at the CTC numbers as a function of time during treatment, PI3K treatment at the beginning. Blue is mesenchymal, red is epithelial phenotype. Initially, lots of CTCs in the circulation. Almost all of them are mesenchymal phenotype. Responds to treatment beautifully. The disease disappears, evaporates. CTCs become almost all epithelial. They dynamically shift their phenotype. Stops responding. You see in real time, you could see that they're not responding, and you change treatment. Responds beautifully again. Suddenly, as it stops responding, it becomes all mesenchymal. Responds to treatment, it becomes all epithelial. And then the patient expired, it was all mesenchymal. So you will call Bob Weinberger was right. EMT is driving the cascade based on this.
Are those days? Are they accepted?
A month.
But the number of CTCs is decreasing.
It decreases.
Yeah. And they're changing their...
And they're changing phenotype. They become more, they're responding, they become more epithelial. They stop responding, the tumor becomes much more mesenchymal. responds again, becomes much more epithelial. So clearly epithelial mesenchymal transition, as Bob Weinberg said, is driving the process. And we have multiple patients like this. What was very interesting is
What was the cancer in the previous one?
breast cancer. And when we looked at these cells, when we saw a lot of mesenchymal phenotype, we saw a lot of clusters. But this chip was not designed to capture clusters. There's mechanical filter, all kinds of stuff. But we saw more clusters. And then we almost saw no clusters when it was all epithelial phenotype. So it doesn't differentiate. From an engineering perspective, the two coupled right now. Because first of all, when it is mesenchymal, the belief is that the cells become epithelial attached to each other, become mesenchymal, it becomes single and move out as single cells, motile. And it's just the opposite of that. So cluster also correlates. More clusters, not good. Less clusters, it's better. Then we looked at patients and see if there's a correlation between survival and the number of clusters. And so we could capture these cells. And if the patient had single CTCs, survival was much better. The red one, if this patient in 3 longitudinal time point had clusters, survival was terrible. So clearly, clinically, breast cancer, it correlated. In prostate cancer, it was even more remarkable. These patients didn't have clusters in their circulation. These patients had clusters in their circulation. So it was both EMT operating and clusters. Then Nicola, one of the postdocs, developed this wonderful animal model. So we wanted to know if clusters are coming out of the tumor as cluster or as single cell and then grow in the circulation, because some of them are proliferating. And that is fundamentally very important to our understanding. So what he did, he took an animal model. He put breast cancer cells. He labeled half of them red, M. cherry, half of them green, GFB. Same cell, no difference, just different colors. Mixed 1-to-1, put it in the animal, and this tumor goes to lung very efficiently. And as you can see, if the tumor metastatic sites or the ones in circulation are 2-color, that means it's coming sloughing off as cluster. If they are single color, that means they're sloughing off as single cells and then growing. We have done many other controls, but this is just the key experiment. When we look at the circulation, first of all, 97.5% of all cells were single cells. Only 2.5% of cells were in clusters, but almost all clusters were dual color. So clearly, they were coming sloughing off as clusters. Then we looked at the efficiency of metastatic cascade. Single CTCs, when we look at the metastatic loci in the lung, we counted how many are single colors, how many are multicolor. If they're coming from a single CTC, it should be single color. If they're coming from a cluster of multiple color, it should be multiple color. And we counted, although 97% of all CTCs were in single cell, 50% of them roughly of the lung were coming from single cells. 2.5% were clusters, but 50% of the metastatic sites were coming from clusters. So if you do the simple math, clusters are about 50-fold more metastatic than single cells. And with an experiment like this, animal model, and a tool like this, you can go after this kind of very fundamental understanding. So that's quite exciting. Then we said, what is in the clusters that is causing this? So we did the RNA sequencing of single CTCs and CTC clusters. Is there a specific difference that's upregulated in clusters? And so we took the single cell CTC clusters and did the single cell resolution sequencing. long story, there was one gene that was 256 times higher expressed, and that was placoglobin. And placoglobin here is part of the desmosome system, cell-cell adhesion. And if you look at the component of desmosomes and adherence junctions, where the cells are attached to each other, this is the adherence, this is the desmosome. Placoglobin is in both of those junctions. Then we wanted to stain for desmoplakin, which is only in desmosomes, and cadherin, which is on the adherent junctions, to see which ones are upregulated. We developed the assays, and when we stain single cells, these are single cells, clusters, nuclear stain, tumor stain, cytokeratin, placoglobin, desmoplakin, and E-cadherin stain, you see that only clusters express those. Single cells did not. Then we went to the patients. We did still more animal experiments. We silenced the RNA for placoglobin. And you see that the metastatic sites were decreased significantly. This is photon count. This is the lung. You see in the silence case, you don't have as many metastatic sites. And turns out to be when we do that, number of cells were identical, but the number of clusters decreased a lot. Then we looked at actual patients, breast cancer patients. And with the primary tumor, we looked at the survival, metastasis-free survival, and those patients that had high expressing placoglobin in their primary tumor and low expressing, there was a difference. So you can see how important these things are, because you can see how you can develop therapeutic targets by knowing these things. And of course, we are working on a number of those. Then one of the postdocs, who's now a faculty member, we said, we are seeing these clusters, but it's like the early days of CTC chip. We don't have the Ferrari version of the cluster chip. We're just seeing, because the chip that's designed for single cell sometimes captures clusters. So we said, okay, can we build a Ferrari version of a cluster chip? And then we built what we call a CTC cluster chip. It will be published next week. So the idea here, unprocessed whole blood comes. We have these individual triangles. And the triangles are designed with a very interesting mechanical feature. It's not size. You see that this is about 12 micron here. That's 12 micron. There are two-cell cluster. There's this apex here. So if a single cell comes, even if it is as big as this, it goes through. And we have done the modeling. For example, here, two-cell comes, it's captured. But if a two-cell comes into a single hole of 12 micron, it will go through it. It's viscoelastic. So the way this works is almost like a cantilever. The forces are balanced such that even a two-cell one, and the flow rate is very small, 70 microliter per minute here. So that's the concept. It's not size. It's the balance of the mechanical forces such that even a two-cell cluster could be captured. When we build the chip, it has 8 parallel units, 8 rows, 4,000 some trapping triangles, and 70 microliter per hour, 33,000 triangles, and 2.5 mil per hour volumetric flow rate. And this is about the size of a business card. And you see the electron EM images of the chip. And look at it. We could capture these clusters. These are from patients. And some of them are huge. These clusters are circulating in these people's peripheral blood. This was we've seen this only once, but it's so beautiful. It's as much as it tried to squeeze by, and it did. Look at this. It went through that little hole, but it couldn't escape. And we have 8 rows, so even if it escaped this one, it will be captured in the next row. And we have done all kinds of studies, but what we now know is about 30 to 40% of all patients, we see clusters. Before it was 5% of the time because we were not going after actual clusters. So it's a very interesting story unfolding, how the primary tumor goes to the circulation, how EMT and cluster biology play a major role, how some of these correlate with the patient outcome and response to treatment, and how these could be, of course, potential target for treatments. We are looking into ways to, of course, disrupt these clusters, and we are looking ways to play with EMT so that we can block the metastatic process in using other approaches. And so what clinically, the part that I didn't talk much today, the way we see this technology play a role at 3 levels. So you have patients, they come, early detection, patient group, treatment selection, and real-time monitoring. So the initial applications are in real-time monitoring. Do they respond? Do they not respond? You look at the number of these cells. You can monitor individually whether or not a patient is responding to treatment. And so this is more in the metastatic patient category. The next level is we take these cells, sequence them, look at the mutations, translocations, so that they're, right now, all drugs that are being developed for cancer is targeted therapy. They are targeting a genetic defect in the cell, in the cancer. There are no chemotherapies that are new that are being developed. All phase 3, phase 2, phase 1 trials are targeted. So from what we call liquid biopsy, you find these cells, sequence it, you can tell which drug to give to a patient. And so that's the near-term, next-level application. Our most longer-term exciting goal is, of course, early detection. Push the sensitivity of the technology to a point where we could detect early. That's the holy grail of cancer. If you could do that, today's treatments are cure for many cancer. And, but the issue there, of course, is the sensitivity and the presence of these cells early enough in circulation. Then it could become a screening modality. In that case, your false positives are intolerable. You cannot look at millions of people and have even tiny amount of false positive and scare the hell out of them. So the sensitivity false positive, you could miss it because other techniques miss it too, but you can't tell them they have cancer if they don't have cancer. And so those large-scale clinical trials are extremely expensive, long time, take long time, and you won't do it until the technology is ready for it. In the very long term, we want to build, from an engineering perspective, point-of-care chip. In the developed world for physicians' offices where you give a blood, immediately you could look at the presence of these, but also a portable one for global health. The data so far is actually such that the number of these and the presence of these cells, forget all the molecular fancy stuff, correlates extremely well with imaging. With MRI, CT scan, and PET scan, even with the earlier less sensitive technology. So if that's the case, if we could reduce this into a proxy for imaging in global health, that could have huge potential. So those are the areas that we are working on right now. With that, of course, this is not done alone. Dr. Haber is the director of Cancer Center at Mass General, is a very well-known cancer doctor and geneticist. And we joined our laboratories, so it is not a collaboration, it is a joint effort. Whatever daily problem we have, we put our money, postdocs, investment, everything in that problem to solve. It's not in collaboration, it doesn't work that way. More like a marriage. You could, and Shamala is a brilliant molecular biologist who oversees all of that. Ravi Kapoor is a wonderful friend. We started a number of companies together, and he joined the team to make it work. So in science, usually we do the discovery work. We are very interested how things work. But if you want to have a real impact, you need to make things work. So Ravi is my most expensive date because making things work is very expensive. most of our money go to making those plastic chips and making it uniformly at that cost factor and whatnot. And that takes real amount. And Shannon does a lot of the science behind engineering. Leisha, lung oncologist, but sees all of our clinical trials and IRB, pathology sequencing, and all the postdocs. Most of the work I mentioned is by Fabio Murat and Emre, the iChip. And Fatih is the cluster chip, but everybody played a major role. Nicola here is the cluster person, and David Miyamoto, all the prostate cancer. And with that, of course, we get funding from NIH to do the basic science, various foundations to do specific cancers for those foundations, stand up the cancer to the multi-scale clinical trial, and Johnson & Johnson has the rights to the iChip, the third generation chip, and they will hopefully commercialize it next year for research use only and then for in vitro diagnostics. With that, thank you for your attention.